Last week, I was tagged in a video of a chiropractor touting the consumption of Schweppes tonic water and zinc supplements as the best way to armor ourselves against SARS-CoV-2 that causes COVID-19. He cited reports of a doctor's success in treating COVID-19 patients with hydrochloroquine and zinc. This is no novel procedure. Many physicians across the globe are turning to hydroxychloroquine and chloroquine as one of many pharmacological interventions in the fight against the new severe acute respiratory syndrome virus. This chiropractor said that we should get our hands on some Schweppes because it has huge amounts of quinine, a very similar agent to hydroxychloroquine and chloroquine. But his Facebook Live got me thinking, why are we using these substances? Quinine has been used as a medical treatment for centuries. It's naturally derived from the bark of the cinchona tree, a plant indigenous to the Andes region of South America. Natives of the region used the bark to treat fevers and nicknamed the plant the fever tree, later giving name to the very popular premium tonic water brand. Quinine's reputation as a treatment for malaria gained traction during colonization. In the 1800s, British troops stationed in India began to consume quinine to prevent contracting the disease, but the substance was so bitter that they required a spoonful of sugar and some water to help the medicine go down. Thus, the first tonic water was created. And because all soldiers kept rations of gin on them, their daily medicinal cocktails became the first recorded G&T happy hour in history. Soon the demand for quinine exceeded the rate of sustainable plant extraction, and a way to synthesize the compound chemically became necessary. In 1943, as a wartime effort, the US launched an extensive anti-malarial research campaign in which thousands of compounds were tested. Chloroquine, one most promising. However, chloroquine, as with quinine, presented many serious side effects. Shortly after, in the 1950s, a hydroxyl group, or an oxygen molecule bonded to a hydrogen molecule, was added to the compound, creating hydroxychloroquine, therefore increasing the solubility of the drug and lowering its toxicity. Yet, unfortunately, by 1980, most strains of the malaria-causing parasite became resistant to hydroxychloroquine and chloroquine. So how exactly do these drugs work against malaria, and how did some species of this parasite become resistant? When a human becomes infected, the parasite first journeys to the liver, where it hijacks liver cells to reproduce. Once the baby parasites mature, they burst into the bloodstream where they go on to hijack red blood cells. Parasites have a stomach of sorts. It's a single structure called a food vacuole. And the food of choice for malaria-causing parasites is hemoglobin, the stuff that is largely responsible for giving blood its red color, but more importantly responsible for transporting oxygen to the rest of the body. When chloroquine is administered, for example, it enters the parasite and it concentrates in this food vacuole. It interferes with the vacuole's breakdown of hemoglobin, and without the nutrients from its foodstuffs, the parasite dies. Yet, over time, these parasites developed functions to eject chloroquine from their vacuoles. So, how did the drug get concentrated in the food vacuole to begin with? Researchers think it's due to a phenomenon called ion trapping. We've all heard of the pH scale, right? It describes how acidic or basic a solution is on a scale from 0 to 14. 0 being the most acidic, 7 being neutral, and 14 being the most basic. Ion trapping essentially refers to the entrapment of acidic substances in basic mediums or basic substances in acidic mediums. Quinine, chloroquine, and hydroxychloroquine are all bases. I know this because I looked it up on Google. But it's also reasonable to assume that quinine is a base because it's what makes tonic water taste bitter. More specifically, these quinine-related compounds are considered weak bases because when reacted with water, they don't completely dissociate, which means they don't completely engage in social distancing. No, actually, it means they don't completely ionize, meaning most of the molecules in the solution don't become charged. A cell membrane is more permeable to non-charged or non-ionized molecules. It's important to note that the space inside the cell is slightly more acidic than the space outside the cell. However, some structures inside the cell are way more acidic, and this pertains to both human and parasitic cells. In fact, the pH of a food vacuole in a parasite is around 4.5. But once a basic non-ionized molecule crosses the membrane into the cell's cytoplasm and into an acidic organelle, like a food vacuole, it does become charged. Once it's charged, it can no longer go back through the membrane passively. This is how basic molecules accumulate in an acidic medium. And this is how an alkaline drug like chloroquine can accumulate in a parasite. As I mentioned earlier, the food vacuole is kind of sorta like the parasite's stomach. So if you want a parallel, you can think of your own stomach. The human stomach secretes a mixture of acids with a pH between 1 and 3 that are crucial for breaking down the food we eat. But say you want to inhibit the action of this acid. It's going haywire, you have heartburn or acid reflux. So you reach for some Tums. You've just fought an acid with a base. In the meantime, chloroquine and hydroxychloroquine have seen a new life as treatment options for autoimmune diseases, HIV, and cancer. But why? The exact mechanism of their action has yet to be elucidated. That's right. After hundreds of years of use and prescription, we still don't know why these particular alkaline compounds work. We only have theories. The same is now true in the fight against COVID-19. There's a lot of hype around chloroquine and hydroxychloroquine, for example, in any of Trump's recent press talks. I expect more research to be churned out soon. And in the spirit of science, I expect everything in this video to be wrong by the time I post it. I'll try to keep the theory simple, but if I've made any errors in understanding, please bring that to my attention. So while I'm not a doctor, I'll take you through how the virus might operate in human cells and how chloroquine might operate against it. We start at the surface, the cell membrane. Although we refer to the virus that lends itself to our current global pandemic, the coronavirus, it actually belongs to an entire family of viruses termed coronavirus. The surface of these coronaviruses contain projection called spike, or S proteins. The scientists who first identified coronaviruses thought the magnified microbe reminded them of the sun's corona, the crown-like ring of gases that surrounds the sun and can be viewed during a solar eclipse. Although it's likely that scientists followed the Latin tradition of naming, simply corona means crown in Spanish. It's no wonder the logo for the Mexican-produced Corona beer brand is a crown. More specifically, it's the crown that sits atop the Church of Our Lady of Guadalupe in Puerto Vallarta, Mexico. Corona beer also sits atop very large shelves at the Puerto Vallarta Walmart just steps away from its cruise terminal. The spike protein is the virus's key to gain entry into the cell. Where's the door? The door is called the ACE2 receptor, angiotensin converting enzyme type 2 receptor. Not surprisingly, it's super concentrated in a type of cell in the alveoli. You know, the little tiny beanbags in your lungs responsible for gas exchange. Once the spike protein locks into ACE2, it's in! Is it really that simple? Scientists believe the door first needs to reveal a lock for the virus's key to fit into. We know that both the spike protein and the ACE2 receptor are proteins that are highly glycosylated. Glycosylation refers to the process of attaching a carbohydrate group, or glycan, to a protein. What results from this process is a change in the shape of the protein and allows the virus to properly bind to the host cell. But where do the proteins get this sugary shape-shifting chain? Where any resident would get its sugar? From its neighbor. That's right. Next door to the ACE2 receptor on the cell membrane is sialic acid, a simple sugar that dangles off the end of more complex sugars and is thought to contribute the properties that help lubricate airways. But you know what also likes sugar? Chloroquine. Recent research posits that chloroquine also binds to sialic acid, disrupts glycosylation, and prevents proper binding of the virus. The end. Or is it? Say chloroquine didn't stop the virus there, because what grants the 2002 SARS-CoV virus entry into a cell isn't a conformational change, but a cleavage of the spike protein. We know that proteins are long chains of other molecules called amino acids. The sequence of a protein's amino acids encodes what the protein is and what it does. But sometimes a protein isn't active until this chain is broken, much like a glow stick doesn't glow until it's broken. Along the sequence of the spike protein is a site for yet another protein, TMPRSS2, to come in and break the chain. But again, we still don't know the exact mechanism of entry. We only know that it sure as heck enters. So viral parts are squirted into the cell. That's over. They're taken in by a structure made up of the cell's own membrane called an endosome in a process called endocytosis. Endosomes are the mail people. They sort and deliver internalized material to other places in the cell. On its journey into the interior of the cell, the endosome matures. It shifts from an early endosome to a late endosome to a lysosome. With each stage of development, the pH inside the vesicle decreases from about 6.5 to 4.5. The acidification of this vesicle has been shown to induce changes in the viral particles that allow them to secretly escape into the cytoplasm, where it can then set up its coffee shop. You remember what other cell structure had a pH of 4.5? The food vacuole and the parasite. And what did we do to curb this vacuole's hemoglobin cravings? Fed chloroquine. The thought is the same here. If you give chloroquine to a virus-infected cell, it will interfere with the acidification of the lysosomes and stop the virus before it even thinks about sneaking out of the house. The end. Or is it? Say chloroquine didn't even stop the virus there. After the viral parts make their great escape, they need to find a place to loot. We'll cut straight to it. Autophagy is the cell's means of cannibalism. Autophagy means self-eat. It's a process in which damaged proteins and organelles within the cell are engulfed by a double-membraned vesicle and disposed of without having to kill the entire cell. Autophagy begins with the formation of a nascent phagophore or isolation membrane. This phagophore matures into an autophagosome, which then fuses with our super-duper acidic lysosome to form an autolysosome. It is here that the particles in the autophagosome are digested by the enzymes of the lysosome, and poof. Coronaviruses, however, learned how to usurp this pathway so that they don't go poof. Instead, they trick the double membrane vesicle into providing them with all the raw materials they need to construct their spawn. Enter chloroquine, which we already know affects the pH in lysosomes. If the lysosomes can't do their whole garbage disposaling, then autophagosomes can't fuse to the lysosome. This leads to a buildup of toxic debris in the cell. The only option a cell has now is to kill itself. No cell to infect means no infection. The end. Or is it? Research shows that although the autophagy pathway is initiated in coronavirus infection, it isn't solely required for success. It probably has a different hideout for replication. So now what? A main reason chloroquine has been used to control autoimmune diseases is because it has documented immunomodulatory effects. That's just a fancy way of saying that it modulates the immune system. How? Well, it could be a byproduct of the mechanisms we've already discussed. All of those processes or structures are instrumental to what is called cell signaling. Cell signaling describes how cells interact with each other, and it's an important phenomenon when considering how the cells and proteins of the immune system are recruited to fight an infection. First, we discussed the activation of the spike protein ACE2 receptor complex. The process responsible for this activation, be it glycosylation or cleavage, initiates cell signaling. This is where the proteins say, Houston, we have a touchdown. But more specific to the touchdown of a viral pathogen is a receiver called a toll-like receptor. These proteins are named not because they look like tolls, but because they reminded the scientists who discovered them of the toll gene found in flies. The scientist who discovered toll genes in flies was German, and toll means great. These toll-like receptors, or TLRs, are great. There are 10 identified types in humans that are found along the cell membrane and on the surface of endosomes. Each type has its own way of recognizing pathogen-associated molecular patterns, PAMPs, but we'll just call them viral parts. TLR7 in particular uniquely recognizes viral parts through its sense of single-stranded RNA. This is significant because coronaviruses are single-stranded RNA viruses. Their genetic material, the only thing that gives them an identity, is a single strand, a lot like our body's own messenger RNA. Once a coronavirus is recognized, it activates the immune system bat signal. Funny because this is a bat coronavirus. And it beckons to macrophages and cytokines and neutrophils and T cells and B cells and all those other alphabetic buzzwords. I'm certain you've heard in recent news reports that it's time to fight. Autophagy, as we've previously discussed, can present viral parts, and more specifically viral RNA, to TLR7 in the endosomes. And this amplifies the production of cytokines even further. If chloroquine interferes with glycosylation and endocytosis and autophagy, it most likely interferes with cell signaling. Thus, chloroquine quells cytokine production and potentially puts a halt to the cytokine storm that unfortunately spirals a lot of severely infected individuals into multiple system organ failure. Now, I'm not going to say the end. There is one more action of chloroquine that brings us back to the beginning of our story when our beloved chiropractor told us to consume copious amounts of quinine and zinc. If the virus managed to set up its coffee shop, it will use an enzyme called RNA-dependent RNA polymerase to make more of its genetic code, leading to more viruses. Zinc is known to inhibit this replication, but zinc is a charged ion. And if you remember back to earlier in this video, the cell membrane is not as accommodating to ionized particles. If zinc was to enter the cell, it would need some help from a transporter, an ionophore. When looking at chloroquine as a potential cancer treatment, a group of researchers inadvertently found that chloroquine is exclusively a zinc ionophore. Chloroquine sensitizes the cells, shuttles zinc into the cytoplasm, and then zinc can do its inhibiting. Not only so, apparently the addition of zinc enhances chloroquine's inhibiting ability. So we should be consuming quinine and zinc. No! While quinine, chloroquine, and hydroxychloroquine are all related, they're not the same. Quinine and chloroquine in particular have a laundry list of side effects and contraindications and adverse reactions with a ton of other drugs. Quinine can be eliminated from the body within 18 hours, but chloroquine may take weeks to evacuate. Clinicians don't even know what the exact minimum dose is for efficacy. And want to know how huge the amount of quinine in Schweppes tonic water is? 67.9 milligrams per liter. The FDA prohibits any tonic water to contain more than 83 milligrams per liter. You'd have to drink well over 2 liters of tonic water a day to even reach a therapeutic dose. I know that while we're in isolation, we have steady access to a toilet, and I'm sure you've hoarded your fair share of toilet paper. But even if this method did work, you'd probably be confronted with a gnarly skin rash before you'd be immune to COVID-19. And in a study that compared the preventative effects of quinine-like drugs, only hydroxychloroquine significantly aided cells in warding off the virus. And because most chloroquine resistance and parasites occurred in areas of high transmission where people received the drug preemptively, I'd be worried about this virus developing resistance before we even had the chance to see what works long term. Don't consume the quinine unless it's with gin and it's for purposes of emotional healing. Except, don't do that either. Recent research suggests that this novel virus suppresses interferons as well. Interferons are the first cytokines that are deployed after the virus is recognized at a TLR site. They are named such because they literally interfere with viral replication. They go on to activate hundreds of other immune factors. But if they're suppressed, then the rest of the other immune fighters aren't deployed in a timely manner. This probably accounts for why so many people present as asymptomatic until a cytokine storm threatens their existence. So if the virus has figured out a way to weaken our innate immune system, your best bet in a preventative measure would not be to weaken your own immune system with alcohol. So to recap, what should you do? Stay home, exercise, eat your vegetables, drink water, bask in the sun. If you're a baller and you have a sauna, sit in your sauna. Strategic application of heat and cold therapies is shown to reduce cytokine storms and bolster the innate immune system. Thank the medical practitioners, first responders, and scientific researchers who are working tirelessly to ensure our safety. And wash your hands. What you should not do? Drink 2 liters of Schweppes tonic water.